We are going to study the electron transport chain or the electron transport system, that is ETC or ETS in short we call it. And this is the third step of respiration. In this step, the NADH2 and the FADH2, which are produced during the Krebs cycle, are utilized to produce energy and water. The process takes place in the inner membrane of the mitochondria, and we are going to understand the process or how the energy release is taking place. Now here I have drawn a diagram where showing a figure showing you the explanation, showing you the structure. This is the matrix of the mitochondria. Since the entire process is taking place in the mitochondria, this is we are talking of mitochondrial matrix and inner membrane, which is having a electron series of electron carriers, which we'll be discussing. And this inner membrane is the site of electron transport chain, or the place where actually the transportation of electron is taking place. This is the outer membrane, and the space between the outer membrane and the inner membrane is known as intermembrane space. Now here we have mentioned over here various electron carriers through which the electron will pass through. Now here the electron flow from high energy to lower energy level. As the electrons will be passing through these carriers, the energy of the electron will be decreased and lowered. And this energy difference will be later on utilized to convert ADP into ATP. Now, here the electrons will be simply passing through the electron acceptors. Let us study about how these electron acceptors are working. Also, it is important for you to understand that we are talking of the electron transport chain taking place in the inner membrane of mitochondria. So these electron acceptors are actually the protein molecules. These are intrinsic protein molecules which are deeply or completely embedded within the inner membrane of the mitochondria. Now, the NADH2, which is being produced during glycolysis over here, glycolysis and Krebs cycle over here, is utilized. Now, NADH2, which is being produced over here, it breaks up into a pair of electrons. Also, it produces protons, and the NAD is regenerated. This NADH2 is obtained by or is produced during the Krebs cycle and the glycolysis process. Now, over here, this electron will be passing through this series of electron acceptor. When we study these electron acceptors are further, for our understanding, we will divide them into complex. So let us divide, there are 4 complexes. This is complex 1. The ubiquinone will be the second complex. Then from here to here will be the third complex, and this will be the fourth complex. So over here we have divided these electron carriers or electron acceptors into 4 complexes, because they are also known as complex 1, complex 2, complex 3 and complex 4. This is for us to understand in a better way how the electrons pass through. Now, the NADH2, which is being produced, breaks up into 2 electrons, that is the pair of electrons, a pair of protons, and NAD is regenerated. this proton, which is being the pair of proton which is being generated, gets stored within the matrix, while this electron will now be taken up by the flavin mononucleotide or FMN, and FMN is the first electron acceptor. NAD is regenerated. Now FMN will be the first electron acceptor. This pair of electrons through FMN will finally reach to the bottom of the chain. Now, this, as electron passes down, it will continuously release energy. When electron passes from FMN to FES, that is complex 1, energy is released over here. Now, when this energy is being released, this energy will be released over here, and this energy which is being released due to the passing of the electron from FMN or the complex 1, it is utilized to pump a pair of protons from the matrix into the intermembrane space. So this energy which is being released over here due to the transfer of electron through complex 1 now will help in pumping the pair of protons from the matrix to the intermembrane space. So we find that energy is released when this, we will mark it as with asterisks, that is showing the release of energy. The electron further moves down the gradient through electron acceptor. So it will pass from here, and as it passes through the ubiquinone, again the energy will be released at complex 2. So here the energy is released again, and this energy is further utilized to pump a pair of protons from the matrix to the intermembrane space. So till now we have 2 times the release of energy which is being utilized to pump the protons, a pair of protons from the matrix to the intermembrane space. The electron, obviously, as we have been as explained, the electron will be continuously losing the energy as it is passing through the carriers. Now this electron will further pass through cytochrome B, and when it again passes through the ubiquinone, it will release energy over here. So energy is again produced at the 3rd complex, and it will also result in the transfer of a pair of protons being transferred from here, that is the matrix, to the intermembrane space. Finally, through the cytochrome C1, C and A3, the electron will be given to half O2. Now we are writing half O2 because we cannot write O. Therefore, we write half molecule of oxygen. That means actually it is 1 atom of oxygen. In this process, when we study, we find that the energy is released by the pair of electrons as it is passing. The pair of electron as it is passing, it is not being utilized anyway. This pair of electron as it is passing through the complexes, it is releasing energy at complex 1, at complex 2, and at complex 3. Along with the release of the energy, this energy being utilized to transfer a pair of protons every time from the matrix to the intermembrane space. But at 4, that is complex 4 cytochrome, there is no release of energy. And the last electron acceptor in this electron transport chain is cytochrome A3. So when we study the last electron acceptor in the electron transport chain is cytochrome A3. And finally, when the electron comes out of the chain, it is accepted by oxygen. So this is a very, this you have to be very clear in understanding, because within the chain, it is cytochrome A3, which is mainly or final acceptor of electron, whereas outside the chain, that is when we are talking, the final acceptor is oxygen. This over here, the utilizing the proton, that is the proton from here, the electrons are already accepted from here, 2 electrons are accepted, then using 2 protons, the water is produced. So the water is generated at the end of the electron transport chain. If we see, in short, one molecule of NADH2, it releases 2 electrons. These 2 electrons, which are released into the matrix, enter the inner membrane, and the first acceptor is FMN. And as it passes through the complex or the series of electron carriers through the gradient, there is a decrease in the energy of the electrons, but at the same time, it releases energy at 3 levels: complex 1, complex 2, and complex 3. Every time it releases energy, there is a transfer of protons, a pair of proton, from the matrix to the intermembrane space. The final acceptor is cytochrome A3 within the chain, and the final acceptor after the completion of the chain, it is oxygen. Now the 3 pairs of protons are produced. Now over here we find, since the protons are transferred every time, the outer compartment will have high concentration of protons. And we know that ions or the particles move from high concentration to low concentration by a process called as diffusion. But this diffusion can only be done through F0 and F1 particles. So this F0 and F1 particles will actually utilize these protons through the transfer to generate ATP, and that is the next step, which is known as oxidative phosphorylation.